anatomists and physiologists. This is another video in our digestive system series. We're going to be talking all about the accessory organs. We'll start by talking about the beginning of the digestive system, the mouth. And remember that digestive tract is just one long tube. The beginning is the mouth, but the mouth has a lot of accessory structures. So even things like the lips, things like the cheeks. They play a role in keeping that food where it needs to be for chewing, and this includes the muscles of the lips and the cheeks. The tongue is also a really important structure for the digestive system to move food around that mouth, to mix it with the saliva, and then the tongue helps make that ball of food and spit, which we call a bolus. So I'll write that word. Um, so it's called a bolus. A bolus is just a ball of food that we've chewed up and moved around with our tongue, coated in saliva, that kind of thing. And what moves that bolus of food from the more front parts of the mouth where we're chewing it down that tube of the pharynx is the tongue. The tongue does all of that stuff. And then the tongue has some bumpy bits on it, which are called taste buds. I'll show you a picture of that. So here's another picture of the tongue. And so all of these little bumps, like all of these little bumps, all of those little bumps, like all of those little bumps are called papillae. Let me highlight that. Papillae. They're just taste buds, different flavors of taste buds. Also here on the tongue, we do have some of our tonsils, which we learned about in the lymphatic system. So, when you're thinking about something delicious and your mouth starts to water, that's how salivary glands work. Saliva is important for doing things like cleaning the mouth, starting to digest those food chemicals. That's what taste buds detect, is dissolved food chemicals. It helps moisten that food, and it also contains the enzyme amylase. Saliva has amylase. Salivary amylase digests starch. And saliva also has a lot of other protections. So saliva has amylase, but it also has protection against microorganisms. So we have things like antibodies. We have things like lysozyme. We have things like defensins in that saliva. Saliva can increase when we activate those receptors in the mouth that send signals to the central nervous system that we're eating. So we have things like mechanical receptors that detect chewing, things like chemoreceptors that detect substances that are in our mouth, like acidic substances, for example, which would lead to a sour flavor detected by our tongue. And this is like the parasympathetic nervous system in action, right? Because that makes us salivate more. Because PSNS, that's this rest and digest one. And there's a bunch of different flavors of salivary gland here. So we have like the parotid glands and those little ducts that release that saliva into the mouth, for example. We have the sublingual gland underneath, sub meaning below, lingual meaning the tongue, so it's the below the tongue gland. We have the submandibular duct, sub meaning below, mandibular meaning mandible, and then the gland that goes with it. Okay, so those are the salivary glands. And then we also have teeth. Well, we get two sets of teeth in our lifetime. The top ones are the baby teeth, so I'm going to zoom in on those. So we have two sets of teeth. The baby teeth, these are the baby teeth. Baby teeth are also called deciduous teeth. Deciduous teeth are That word deciduous means that it falls out. So trees that are deciduous are the ones that lose their leaves in the fall. Another word for baby teeth are milk teeth, or we can also call this the primary teeth or the primary dentition. Dentition. So then we lose those in childhood, and those baby teeth, those primary teeth, get replaced by the permanent teeth or the permanent dentition or your adult teeth. But notice that there's a lot of big similarities here. We have a lot of similar types of teeth. The other thing here is that the front teeth and the back teeth look a little bit different. So here are the front teeth, here are the back teeth. So the front teeth, they're called incisors. Incisors are sharp and they're good for cutting. And then we have the canines, the canine teeth, or the cuspids, or the eye tooth. Those are really good at tearing. Then we have premolars or bicuspids. Premolars or bicuspids, those are like these guys, they have two cusps. And those are really good for grinding along with the molars. So the molars, these are those back teeth, the flat ones. And again, good at grinding and crushing. So each of these is helpful when it comes to eating because they do a slightly different job when it comes to actually doing the biting off and the chewing of the thing, the mastication process. So what does a tooth actually look like? And this is a canine tooth, what we're looking at. This is a section of a canine tooth, but generally teeth look like this, like a general structure. Okay, so the tooth is situated in the socket in the bone. That's the bone there. And the bone is either the maxilla, the upper jaw, or the mandible, the lower jaw. Um, and the part of the tooth that you can see is called the crown. The part embedded in the bone, that's called the root, and then that part in the middle, that's the neck. And the neck of the tooth is typically covered by the gums, which are called the gingiva. So, like gingivitis is inflammation of the gingival tissue. And then the crown of the tooth is covered by enamel. So, the crown of the tooth is covered by enamel. Enamel is super, super strong and super mineralized. The thing that's great about enamel is that it's really strong. But the thing about enamel that's not great is that when it's gone, it's gone. We can't really replace it. Um, And so there's the enamel, the hard stuff. Underneath the enamel is the dentin. So it's all this blue stuff. Dentin is a really great shock absorber. It's a lot softer than enamel, but it's more flexible. So they kind of work together. And then the deepest part of the tooth is called the pulp. So there's the pulp cavity with the pulp, blood vessels, nerves, that kind of thing. And so what can happen is dental caries. Dental caries are cavities. Um, is when we get like a little deposit of bacteria that works its way, damages that enamel, eats it away, and then we can eat away at that dentin. And then eventually, if we get down into that pulp, that's when we start to get really, really painful dental caries, and we might need things like root canal or even extraction of that tooth. So that's the teeth. And all of the accessory structures of the mouth, I think I wanted to tell you about. But after the mouth, the next accessory organ that we're talking about is the liver. That's what we see here. The liver sits right below the diaphragm, and it's kind of situated inside of the rib cage. So let me zoom back out. And the liver is located a little bit like on the right-hand side, but it's super, super big and it has a lot of jobs. But for this unit, we're just going to talk about its digestive roles. And what it does is make bile. So the liver makes bile. Liver makes bile. And we'll talk more about that in a sec. But the liver, it's divided into four lobes. We see the two of the lobes here. We see the right lobe on the right and the left lobe on the left. So the right lobe is a big one, left lobe a little bit smaller. And this is from the anterior view. But if we were to flip that liver around to the posterior, this is what we would see. So we'd see the backside of the liver. And here we can still see that right lobe on the right and the left lobe on the left, but we're on the posterior side, so it's backwards. But we also have the two other lobes here, the two of the four lobes. We have the caudate lobe, and we also have the quadrate lobe. So the caudate lobe, that's this one on the top, and then the quadrate one is that one on the bottom. This is a cartoon image of the same thing. What I want to point out here is that we can also see the falciform ligament. The falciform ligament is actually a mesentery, so it's a double layer of peritoneum, which is just a serous membrane. And that falciform ligament separates the right and left lobes. The liver is also covered by visceral peritoneum, except for this part called the bare area, because it's bare. We also have a structure called the porta hepatis. The porta hepatis is really interesting. It means gateway to the liver, like porta, portal. And that's just the joining of the hepatic portal vein. Oops, zoom back here. So we join the hepatic portal vein and other veins to enter the liver. This is the same view of the liver that we are just looking at. But note that you can also see this little green one, and I'll even highlight it in green for fun, is that we see the gallbladder. The gallbladder is this little green balloon-looking thing. The gallbladder is not part of the liver, like it's its own organ, but it's closely related to its digestive functions, which brings me to what does the liver do? And it makes bile. So we have to think about what bile is. So bile is a fat emulsifier. Which means that it breaks up fat into smaller bits, fats like lipids, into smaller bits. So how I think about this is if you've ever seen like a vinaigrette salad dressing, it's like vinegar and oil, and you've ever seen it sitting for a while, it's like two layers, right? There's one vinegar layer, whatever it is, and then one oil layer. Then before you pour it on your salad, you're supposed to shake it up and mix it before you put it on your salad. And then it like becomes one thing again, right? But if you were to look really, really closely, there's like little balls inside. And those little balls are little balls of fat or oil. That's emulsifying. And what this does is it makes fats easier to digest. Fats easier to digest. And that's important because fats are really big molecules, so breaking them up is a really, really necessary task. Okay, so then once the liver emulsifies the fats, it goes into the gallbladder. So then the gallbladder is bile storage. And we'll talk about, there's like a bunch of ducts here, and we'll talk about all of those. Okay, so back to the liver. We're going to talk about the liver. So in your brain, flip the liver around. So now we're looking anterior view of the liver. We've taken a cross section of it, and I think the liver is one of the coolest organs, histologically speaking. It just looks so cool. So we've taken a cross section of the liver and we've looked at it on a microscope. So all of these hexagons, those are little units of the liver. Those units of the liver are called liver lobules. So each of those hexagons is a lobule. And in real life, these are small, like a sesame seed size. And if we zoom in on that more, we can see there's a little like dot in the middle, and that's the central vein. But there's a bunch of connective tissue connecting one hexagon or one liver lobule to the other. So let's look at a cartoon picture of just one liver lobule, which is just this one here. So this is just one liver lobule. So notice we've taken from the liver one of these hexagons, zoomed in, and then this is a cartoon version of that. The first thing to note is that there's a lot going on in here. I'm going to zoom in because I'm going to be able to talk about it. Okay, there's a lot going on here, but it's all like blood flow and stuff. Okay, the first thing is all of these little like orange cells, those are all hepatocytes. Hepatocytes are liver cells. So all those little orange guys are liver cells, hepatocytes. And they're kind of stacked like bricks, right? And then this middle hole is called the central vein. And notice the direction of blood flow here. So we're leaving that liver lobule. And then each of the points of that hexagon are these like groups of 3 vessels. And a group of 3 things is called a triad, and this is part of the hepatic portal system, so we call it the portal triad. And so each of those 3 things, so we have the bile duct is first. The bile duct is this green one. The bile duct contains bile. Bile is made by hepatocytes. So I'm going to write this. So bile duct contains bile, which is made by hepatocytes. Those are the orange cells. And how the bile gets there is by traveling through these little tiny canals called bile canaliculi. That word canaliculi literally means tiny canals. So all of this little stuff here, all of those little tiny vessels, tiny bile canals. And so that bile will be made by those hepatocytes, travel through those bile canaliculi into that bile duct, and then out. Bile is pretty alkaline, which is basic, and it contains a lot of stuff, but the digestive stuff that we care about are bile salts. So bile contains bile salts and phospholipids. Bile salts and phospholipids. There's also bile pigments in there, which we'll talk about in a little while. Bile salts, these guys, bile salts, they come from cholesterol and they stay in the liver. So we don't really remake them all the time. We actually recycle them a lot of the time. And I'll show you a picture of that in just a minute. Okay, so back to the portal triad. So back to the portal triad, we have the bile duct, we've covered that. The portal arteriole that brings oxygen-rich blood from systemic circulation. And then lastly, the portal vein brings nutrient-rich blood. So the portal venule nutrient-rich blood from GI organs. Do you remember I told you in blood vessels how the hepatic portal system is that stop that all of the blood from the digestive organs makes before it heads back to the heart? Yeah, it's here. Now we're talking about it. This is why. It's to stop at the liver, do the bile thing. I want to discuss a little bit more about these hepatocytes, these little orange cells, though, because they do a lot of things, okay? Yes, they make bile, but they also play a role in glucose storage as glycogen. They make plasma proteins, they store vitamins that are fat soluble, they play a role in detoxification. So they're really, really important, those hepatocytes. The liver does a lot of things. Okay, so lastly, at the portal triad. So we bring in blood that's oxygen rich and nutrient rich, and then we pass through what's called liver sinusoids. So those sinusoids are just extra leaky capillaries. And then finally, that blood leaves through its central vein, and then we enter into the hepatic vein eventually. Okay, so that's the liver. Super cool, super weird. This picture is showing you the steps of how bile salts are recycled. Remember, I told you they're recycled, right? We don't remake them all the time. And it's called enterohepatic circulation. That's what this table is, or this figure is called. Enterohepatic circulation. Hepatic means liver, entero means gut. So it's gut liver circulation. And it's exactly that. It's blood flowing through the liver. So blood going into the liver. And in that gut allows us to conserve and recycle those bile salts. So the first step is the secretion of the bile salts into the duodenum of the small intestine. And remember, I told you that most of that digestion and absorption happens in the duodenum. Yes, it's still the same, okay? But then lipid digestion starts. So then lipid digestion starts, and then we can start to absorb that, and it's all aided by the bile because it's the emulsification thing that's happening. And then a lot of those bile salts, 95% of the bile salts, get taken back into the blood in the ileum. So there's the ileum. So from the ileum, we take those bile salts back into the blood and then send it back up to the liver. So that means that we only need to remake 5% of bile salts. So we're recycling, right? The main bile pigment, so bile pigment is called it's called bilirubin. Bilirubin. And that's a byproduct of red blood cell breakdown, which the liver also plays a role in, but that's a side story. Bilirubin is released into the bile, and then it gets metabolized by the gut flora. Bilirubin can get broken down into stercobilin, which gives feces that brown color. Bile is really important for fat breakdown. So if we didn't have bile, fat breakdown wouldn't happen. And if that doesn't happen, then our feces would appear white because those fats in it, right? And there'd be no bilirubin, thus no sterobilin, and then definitely no fat breakdown because we wouldn't have that emulsification thing. The fats are just too large. The liver is important beyond digestion for metabolism and stuff, so it's really necessary. So if it's inflamed, that's called hepatitis. Hepatitis is a really general term for inflammation of the liver. And there's lots of possible causes, and some examples I've included here. So it can be like things like a virus, and they're abbreviated, right? So it can be like hepatitis virus A, hepatitis virus B, HVC, HVC, that's hep C, or hepatitis C. That's one of the most common versions, and that's the one that's kind of most rampant, and that's done by being transmitted through blood. But some of those hepatitis viruses can be gotten by eating like contaminated food. Another cause of inflammation of the liver, hepatitis, is alcohol, drugs, or wild mushrooms, and chronic alcohol abuse or anything like that can lead to more severe forms of hepatitis, more chronic hepatitis, which can lead to more of a disease state. Interestingly, we can also have a non-alcoholic cause of fatty liver disease, and that's called NAFLD, which is increasingly common. It's the most common form of liver disease in North America, according to our book. And the risk factors include things like obesity, insulin resistance, and because these things tend to accompany lipid metabolism problems. There's not really symptoms associated with this disease necessarily, but we can then go into more advanced stages of chronic inflammation, liver disease, that kind of thing. So, speaking of that, cirrhosis is the most severe form of liver disease, which can sometimes be a result of chronic hepatitis due to things like alcoholism, this NAFLD, um, other forms of hepatitis, that kind of thing. The thing about the liver, though, is that we can regenerate it in healthy folks. And even in folks with cirrhosis, we can still regenerate those cells, but hepatocytes that get damaged can also make scar tissue. And making scar tissue takes up space that those new and healthy hepatocytes could occupy. So over time and over time and over time, the liver is going to become more and more fibrous and less functional. And it can even mess with the portal system blood flow, and that can be called something. Here it's called portal can lead to portal hypertension, which is when that blood flow is interfered with at that portal system because the liver is just less functional. Um, and cirrhosis is usually treated with a liver transplant, but the organ donation part, this is the really fascinating thing. Organ donation of the liver is cool because we have such a great regenerative capacity for the liver, especially in a healthy person. So if we donate part of our liver, we can remove a portion of it and then that'll regenerate within like a year or so. So cool. Okay, so back to this posterior view of the liver because we need to talk about the gallbladder a little bit more. So, the gallbladder. The gallbladder is really squishy because it has pretty thin walls and it's bile storage, right? And it expels bile through its ducts, and then that goes into the small intestine. But you may have heard of gallstones. Gallstones are little crystals. So gallstones, and gallstones are just crystallized, crystallized cholesterol. This can happen if there's either too much cholesterol in that bile or too little bile. So if it's just the balance is out of whack. And those gallstones can be really, really painful. They can be dissolved with medications or treated in other ways. Like now we can break them up with vibrations by vaporizing them, or even surgical removal of the gallbladder. But the thing is that if that flow of bile from the gallbladder is obstructed because there's a gallstone there, um, this can lead to bile pigment starting to get accumulated in the blood. So bilirubin is that main pigment, right? So bilirubin can make it into the skin, and then the skin starts to appear yellowish, and that's called jaundice. So jaundice, and that's yellowed skin due to accumulation of bilirubin. Jaundice can also be a sign of liver disease, so something to watch out for. Okay, so we've discussed the accessory structures of the mouth. We've talked about the liver, we've talked about the gallbladder. And then the last thing that we need to talk about is the pancreas. The pancreas has both exocrine and endocrine functions, but here we're talking about the digestive parts, which is to make enzymes that break down foods and send them to the small intestine. The pancreas, so makes digestive enzymes. And then they send it to the small intestine. Pancreas is mostly retroperitoneal, which means that the backside of it is not covered by peritoneum. Okay, so what we're looking at here is this is a zoomed out version of the pancreas, and this is zoomed in on some of the important functional parts. So the exocrine part of the pancreas makes pancreatic juice. So exocrine makes pancreatic juice. This duct that we're looking at here, this duct that we've zoomed in on, and that part of the duct as well, it's what we're zooming in on. Okay, so this is a zoomed in part of like this little tube with these little branchies. So we're zooming in on those. Okay, so this word, the plural version of this word, acinus, is acini. It's a plural. But in these little guys are acinar cells, acinar cells. Those acinar cells are the ones that are making enzymes. Lots and lots of digestive enzymes. They're digestive. The digestive enzymes, they get released from the cells into ducts, and those ducts are lined with duct cells. So we have acinar cells and we have duct cells, and the duct cells also release bicarbonate. So the enzymes I'm highlighting, and then also bicarbonate, and a little bit of water too. But bicarbonate is alkaline, and so that helps neutralize that acidic chyme that'll be coming into the small intestine. So speaking of these enzymes, some examples of those enzymes are proteases. Proteases digest proteins. Digest proteins. Amylase. We've met that person before. Amylase digest starch. Lipase. Bunch of different types of lipases. Those digest fats. And then lastly, we have nucleases. Those guys digest nucleic acids, which are DNA, RNA. But the thing is here is that if we were to make all of those enzymes here, release them into those ducts, that's a lot of stuff that we're digesting. And so if we released all of those enzymes into the pancreas, into this little duct of the pancreas, we'd start to digest the pancreas tissue. And so the enzymes that are made in the pancreas are actually the inactive forms. Think like the ogen forms, the generating forms, the precursors, so that the enzymes don't digest the pancreas. And then within the small intestine, then we have mechanisms to activate those enzymes. Let me show you what I mean. This is a specific example that I don't necessarily care that you know. What I care that you know are that once we get from the pancreas, that is our ogen form, our inactive form. So from the pancreas makes inactive enzymes. Once we get to the small intestine, The small intestine is the one that activates the enzymes. So in this specific example, we see that we've released this precursor, this inactive form of an enzyme called trypsinogen, and then we can make an active form of the enzyme trypsin and a bunch of other ones that end up being active there at the end. So we can make trypsin and chymotrypsin, carboxypeptidase, that kind of thing. Again, specific example, but inactive enzymes from the pancreas activated in the small intestine is what I'm looking for. Okay, so the liver makes the bile, the gallbladder stores it, pancreas makes digestive enzymes and makes the whole thing alkaline. But how does all of that get into the small intestine? How do we get there? And it's a bunch of ducts. And it all ends up together. So starting at the liver, we have the right and left hepatic ducts. So we have the right and left hepatic ducts, right, left hepatic ducts. They join together to form the common hepatic duct. We saw a convention like this when we looked at blood vessels. When there's a right one and a left one, or an external one and an internal one, they join together. That's the common one. And then from the gallbladder, the one leaving there is called the cystic duct. The cystic duct and the common hepatic duct merge to form the bile duct, because that's exactly what's in there. It's bile, the bile duct. From the pancreas, so now switching over to the pancreas, we have two ducts. I'm going to talk about the main one. So the main pancreatic duct is called the main pancreatic duct. And then the main pancreatic duct merges with that of the bile duct. So we have the bile duct, main pancreatic duct, and it joins together at this little like volcano-shaped thing. And that's called the hepatopancreatic ampulla. Hepato means liver. Liver. Pancreatic means pancreas. Ampulla is a new word that means flask. Like a collecting chamber, okay? So this is the passageway from those ducts. We've merged them together into the small intestine. And that happens by passing through the major duodenal papilla, a nipple-like structure. It's just the passageway. There's also another pancreatic duct called the accessory pancreatic duct, which is just smaller and it has its own pathway. So it's smaller, the accessory one is just smaller and enters its own way. Note here that there's lots of sphincters. There's a sphincter there, a sphincter there, a sphincter there, and that's to regulate the entrance or not entrance of all of those products into the small intestine. This is controlled by hormones released by enteroendocrine cells. So there's two new ones I want to introduce you to. So there's hormones. And remember, these are made by enteroendocrine cells. In this case, they're made by the duodenal enteroendocrine cells. Okay, so there's two new hormones I want to introduce you to. One of them is called cholecystokinin, or CCK. And then the other one is called secretin. CCK is made by those duodenal enteroendocrine cells, and it's released in response to proteins and fats that are in the chyme entering the small intestine. So from the small intestine, or from the stomach, chyme enters duodenum. If there's lots of proteins and fats, then that releases CCK. CCK acts on acinar cells to release their products. It encourages gallbladder contraction. And it relaxes the hepatopancreatic sphincter. Those are three really different sounding jobs, but all of those things work to put digestive stuff into the duodenum. Okay, so the second one is secretin, still made by duodenal enteroendocrine cells, but it's released in response to the acidity of chyme. So chyme is really acidic because the stomach is acidic, and secretin causes bicarb to be released from duct cells of the pancreas. And bicarb works to neutralize the acid of the chyme, because while the stomach is acidic, the small intestine is not. Okay, so here's what I mean. This is a summary of all those steps that I just talked about. This is in a stepwise fashion. I don't really care if you know the steps, but it is a nice picture to show you like what happens and where. And the colors here, the red is CCK and the yellow is secretin, so you can see where it acts. And this is the last slide for this video. Yay!